This video presentation is on the process of FPLC purification, which stands for Fast Protein Liquid Chromatography. This process was used preparatively to separate MGH from contaminating proteins produced during overexpression. Preparative methods are those used for isolation purposes and involves the use of the entire sample. During overexpression, E. coli cells containing the MGH plasmid were spread onto a cell plate that is used for separation. A colony of cells was then placed in a broth that allowed for increased cell growth to a certain density that can be measured at A600. This produces an uninduced cell culture that is grown for 12 hours in a temperature of 37 degrees Celsius, which is an optimal temperature for E. coli growth. The cells were then incubated with IPDG, which is a compound used for overexpression. This creates an induced cell culture that is then placed in a temperature of 16 to 20 degrees Celsius, which is used to slow the growth and activity of the bacteria. This low temperature allowed the protein to fly under the radar and prevented bacterial precipitation of the protein. After the incubation of the induced cell culture, a cleared lysate solution was prepared. The enzyme lysozyme was used to weaken the bacterial cell wall. Sonification was then used by utilizing sound waves to mechanically break apart the bacterial cell wall. This allowed the contents of the cell to spill out. The compounds DTT and PMSF were then added to the solution. DTT is a thiol compound that breaks disulfide bonds. This is useful to prevent our protein from forming bonds with other proteins in solution. PMSF is a protease inhibitor, which is useful because it limits the amount of our protein that is degraded by bacterial proteases.
MGH is our protein of interest that we want to separate from other proteins. The M stands for malate dehydrogenase, which is an enzyme that we will study later by performing kinetic experiments. G stands for green fluorescent protein, which is used to track the progress of our protein as it turns the sample a bright green color. H stands for the 6 histidine residues, which is helpful for purification. It is unlikely that other proteins will have 6 sequential histidine residues. In order to purify this protein, affinity chromatography was used with nickel NTA column. Nickel has a high affinity for histidine, which causes the protein to bind to the column. After the MGH binds to the column, all other proteins pass through. The MGH can be eluted from the column using imidazole. Imidazole is similar in structure to histidine, so if the protein comes off the column briefly, the imidazole will bind in its place. The MGH can no longer bind to the column, so it must continue through. During the FPLC process, 3 solutions were used. The first was the lysis, which was a cleared lysate solution in a low salt environment. We also did not want the column to contain EDTA, as this compound would inhibit the binding of histidine to the column. The next solution used was a load buffer, which contained high salt concentrations and low imidazole. The low concentration of imidazole was used to elute any proteins that had slight affinity for the column. This concentration was not high enough to elute the MGH sample because our protein has high affinity for the nickel NTA column. The last solution that was used was elution buffer, which contained a high concentration of salt and a high concentration of imidazole. The large amount of imidazole will elute the MGH sample from the column by binding in the place of histidine. Each of these solutions also contain DTT and PMSF.
2 lines can be seen on the FPLC trace. The blue line expresses the amount of total protein that is present. The red line expresses the conductivity of the sample, which refers to the amount of ions in the solution. As seen on the graph, the red line stays at the same level for the majority of the time, but briefly dips lower as the lysis buffer passes through the column. The lysis buffer contains no salt, so it has a very low conductivity. On the graph, the blue line has 2 regions where it contains large peaks. The first peak could be due to 3 possible scenarios. The first scenario is that the protein moving through does not contain histidine, and therefore has a very low affinity for the column. This scenario would be the most favorable because it would mean that our sample remained bound to the column as other contaminants traveled through. The second scenario could be that the column was overloaded. The column has somewhat of a seating capacity, meaning that if the spots in the column are full, nothing else can bind. That means that our protein could be bound, but might not be if the column was already full. The last possible scenario is that the protease activity could have cleaved the bond between the green fluorescent protein and the histidine tag. This means that the histidine tag would be bound to the column, but the rest of the protein would flow through. The second peak indicates that a protein eluted through the column after the addition of imidazole. We cannot definitely say that the peak is due to MGH because of the scenarios mentioned previously, but it is likely that the protein contains a histidine tag. Based upon the peaks seen on the graph, we recommend that fraction 3 is analyzed to determine if any MGH is present in with the other proteins. It is also recommended that fractions 18 to 23 are pooled because that area contains the greatest sample of the second peak, which we hope contains the MGH protein. The spectroscopy team will refine our suggestions.